Okay, so this is all about biotechnology. There's a lot of little things you need to know here. Some of this you know from Project Lead the Way. I know depending on the year that you're watching this, maybe you weren't able to do as many labs as you had hoped, but hopefully you at least got some experience with some of these things in one of your Project Lead the Way classes. All right, so the first is I can define genetic engineering. All right, first of all, what is an engineer? An engineer is somebody who solves problems. So if you are a mechanical engineer, then you solve problems dealing with mechanics, so how things work. If you're a bridge engineer, then you figure out how to build bridges that are stable and will hold lots of cars and whatever. And also deal with weather changes, all that kind of stuff. So when we talk about a genetic engineer, what they are doing is they are trying to solve problems in the genes or the genome. And so they are manipulating the genes, they're changing them, they're tweaking them, fixing them to try to fix and solve problems that we have. And sometimes those problems are diseases. So we're always looking for ways to tweak the genes to try to fix a genetic disorder. And sometimes it's something like increasing crop yield. Maybe we have a bunch of bugs eating our corn. And so if we can create a pesticide that the corn produces itself, kind of a natural pesticide that kills the bugs and doesn't hurt people, then we can increase our crop yield. So sometimes it's things like that. But that's what genetic engineering is. It's basically changing the genes or genomes of organisms to solve some kind of problem. All right, I can explain how a restriction enzyme works. One of the big tools that we use in genetic engineering is restriction enzyme. Now, restriction enzymes are enzymes, so they are catalytic molecules that are going to cut DNA at specific places, and that's where the restriction part comes in. They are restricted to a particular place in the genome. So, for example, let's say this is a section of DNA, okay? And I'm going to cut it with EcoR1. EcoR1 is an enzyme, a restriction enzyme that comes from E. coli. So they're always named after where they come from. And it cuts at between, it's going to look for GAA-TTC. Okay, so every time it sees that, it's going to cut between the G and the A. It's going to break the hydrogen bonds between the A's and T's. And then it's going to cut the sugar phosphate bond between the A and the G. So it's going to cut that kind of stair-step cut. And so it's going to scan, scan, scan. Oh, there it is again. It's going to cut there. Okay, and so it's going to produce this fragment over here, however long it is, this fragment, and then this fragment, however long this fragment is. Okay, because I'm assuming this DNA goes on each end. Okay, and so what we would get is we would get 3 different fragments from 2 cuts. This is really useful to us because it's going to create what we call sticky ends. So you can imagine, if I had scissors, or if I could do this quick enough, if I could cut this, let me get sideways so I can kind of hide this, okay? What you have here is you have this fragment has this little piece sticking out that's TTAA, because this down here is gone, okay? That's called a sticky end because those A's and T's, they don't want to be out there by themselves. So they're going to look for an AATT to bind with. So let's say that let's say this is the insulin gene. Okay, I cut it out using EcoR1 and I have these sticky ends. And then I go and find a plasmid from bacteria and I cut it. I find the same sequence in it and I cut it with EcoR1 as well. Well, when I open that plasmid, it's going to have matching sticky ends to my gene of interest, this insulin gene, so that when I put them together, they're going to want to bind because this TTAA sticking out on the insulin gene is going to match with an AATT from the bacterial plasmid because, again, it cut at the same location because EcoR1 only cuts there. So that means that these sticky ends that are sticking out here and here are going to match the sticky ends on my plasmid when I cut it and open it up. And so that's the benefit of using a restriction enzyme is when you cut 2 different genes with the same restriction enzyme, you're going to get matching sticky ends, and so they're going to be able to fuse together more easily. Okay, so I can explain how a restriction enzyme works. I can explain the purpose of a vector. A vector is anything that carries something from point A to point B. And so what we want to do is, let's say we want to get this insulin gene into this bacteria over here. Okay, but I can't just plop it in there, okay? So what I'm going to do is I'm going to cut it out. I'm going to cut open this plasmid. I'm going to insert this gene into the plasmid, and then I'm going to encourage the bacteria to take up this plasmid. That makes the plasmid the vector, because the vector is taking the DNA from point A out here to point B inside the bacteria. And once it's in there, the bacteria will start transcribing and translating it and making that insulin. I can define transformation. Transformation is the process of bacteria taking up foreign DNA. So this is like a plasmid. And we can encourage bacteria to do this. We know what conditions they like to be more receptive, and so we can encourage bacteria to do this by giving them heat. When you give bacteria heat, it creates pores in their membrane. We can also use certain chemicals to stick the plasmid over here. So when the plasmid is stuck to the membrane and then a pore opens up, it can just right into the bacteria. But transformation is that process of bacteria taking up foreign DNA, and it's called transformation because it can cause the bacteria to do things differently. It can transform it, make it act differently. We use this to our advantage, like for example with insulin. We can have the bacteria make insulin for us. This is how we used to get things like insulin, human growth hormone. Okay, for people who didn't have human growth hormone, we could make it in the lab, as well as factor 8. Factor 8 is a clotting factor that hemophiliacs are missing, and so we would get bacteria to make that for us, and then we could use that to treat hemophilia. All right, the next process is PCR. So I can explain the process of PCR. So here we have our double-stranded DNA, okay, that's stuck together, right? These are little hydrogen bonds in here between the bases. What we're going to do with PCR, PCR stands for polymerase chain reaction. Polymerase because we're going to use polymerase to do more DNA, right? You need DNA polymerase to make DNA. And then chain reaction because it's going to be like a set of dominoes. Once we start, it's just going to continue on. So what happens is we go through this heating process, and I don't remember, but it's like 90 degrees Celsius or something like that. It's really hot. And that heating is going to split our DNA apart. Okay, and so that's called denaturation. We're going to denature the DNA. We're going to change its nature. Okay, and so that's done with heat. And then we're going to cool it down enough for these RNA primers to bind. Right? Remember, you have to use RNA to build new DNA. You can't just build new DNA. And so we cool it down enough to allow that to happen. Okay, and that cooling step is called annealing. To anneal is like to stick something to something else, and so when we cool it, it causes these primers to stick. Then we bring in DNA polymerase. Okay, and we're going to use a special one. We're going to use Taq polymerase. Taq is a bacteria, Thermus aquaticus, that can stand really high. Actually, they live in hot springs, so they're adjusted to really hot temperatures. So when we go through this heating stage, it won't kill this polymerase. So it's just a specific species of polymerase. Everybody's polymerase is a little different. Okay, we have Homo sapien polymerase. They have a different one because they're different than us. And that DNA polymerase is going to attach, and then it's going to start building. I'm running out of colors here. It's going to start building this new strand of DNA. Okay. And of course, it's going to build in the 5' to 3' direction. Okay, we're going to do that again and again. And so what you would see is after one round of this, you have 2 strands. Then those are going to split, and you're going to make 4 strands. Okay, after round 2, and then after round 3, you're going to have 8 strands, right? Because both of those are going to give rise to 2 different strands. Okay, and so what you would expect to see in a perfect world is it would go 2, 4, 8, 16, 32, 64, 128, all that. It's not a perfect world, so we don't always get, like maybe this one, something happens and it messes up, so we don't get that one. So we actually only got 7. But still, after a couple of hours of doing this, and you do these cycles really quick, I mean, they only take maybe, I don't know, 5 or 7 minutes to do one full cycle. So after a couple of hours of this, you can have thousands or even millions of strands of DNA. This is really beneficial because it allows us to make, let's say that you have a little bit of DNA under the fingernails of a murder victim. And it's not enough to do a bunch of tests on him. We need to do a bunch of tests to figure out who's guilty. We can do PCR to make bunches of copies of that DNA, so now we can run a bunch of tests on it. And that allows us to learn more about that DNA and hopefully find the suspect. All right, I can explain the process of gel electrophoresis. Gel electrophoresis, whoa, that's an ugly gel, pretend like it's square, is where we're going to drop the DNA fragments. We've cut it with restriction enzymes, so it's different fragments. We're going to drop these into wells, so these are called wells. We're going to attach electrodes, so negative here, positive here. DNA is a slight negative charge, so it's going to be drawn towards the positive because opposites attract. But the problem is when you look at this gel from the side, so let's kind of like squat down and look at it from the side. So here's the top, here's the bottom. Here's the negative end, here's the positive end. What's actually in here are these little molecules of agar. Okay? And so as your DNA is passing through from the negative to the positive, it has to kind of navigate around all of those. And so the bigger it is, the harder it's going to be to pass. And so it's going to slow it down. If it's a little piece of DNA, it's going to zoom, 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 zoom. So that's why we see this forms bands. And so down here at the bottom, you'll have smaller pieces of DNA, so smaller pieces will go farther, and your larger pieces will be closer to the wells. And so it separates it out. And then you usually run a ladder, which is just, and it looks, the reason it's called a ladder is because it looks like a ladder. And these pieces, these bands that the ladder produces are of known sizes. So maybe that's 200 base pairs, maybe that's 500 base pairs, maybe that's 700, 1,000, or whatever. And so we know because this band is just below that, it's probably in the 450 base pair range. And so that ladder just tells us that. So that's the purpose and process of DNA sequencing. Cloning, I'm sorry, that was gel electrophoresis. DNA sequencing is where you're going to add fluorescent dyes to DNA. Let me find it in here because it'll be a whole lot better than me trying to draw it. Oh, I don't have a picture. But with DNA sequencing, you code each color or each, sorry, I can't type and talk at the same time. You code each base with a different color. And so, for example, in this one, A was coded with green. There we go. T was coded with red, G with yellow, and C with kind of a purpley blue color. And so as it passes through the machine reads the bases, and it can tell you the order of the bases. And that's really important for identification of diseases. It also helps us with evolution, so we can see how different your DNA is from, say, a dog, and that tells us a little bit about the evolution. All right, the process of gene cloning, and I got to speed it up. I got about 2 minutes left, is we take a somatic cell, so it has all the chromosomes. We take an egg cell, we take out the nucleus because we don't want those chromosomes. We take the nucleus from the somatic cell and put it in the egg, or we just fuse the two together. So what we have now is an egg cell, which has all the things we need to divide, and we have the full complement of chromosomes. For us, that would be 46. And then we put it in a surrogate, and we let that surrogate give birth to that baby. And so the baby is going to be identical to the somatic cell donor, okay? So this sheep. So that's the process. A stem cell is a cell that can give rise to any other kind of cell. It is not specialized. Totipotent stem cells can give rise to anything. And then, sorry, I was looking at my time. I got a minute and a half. Totipotent is the most powerful. That's what it means, totally powerful. It can give rise to anything. As they start to shut genes off, it goes from being totipotent to pluripotent, from pluri to multi, and multi to uni. Unipotent means it has one power, has one job. It's just a neuron. It's just a muscle cell. It's just a, whatever, a pancreas cell. And so stem cells go through that process by turning genes off. And then the last thing is applications. What do we use DNA technology for? And I mentioned this a little bit earlier. Agricultural applications, big time. Genetics with diseases and treating those diseases, big time. Making drugs. We talked about that with transformation and making insulin. Also in forensics. But also we use it to clean up the environment. And these are just a few. There are many, many other ways we use DNA technology. It is a big field. It has a lot going on. It has been so helpful. And this has really all blossomed in the last 50 to 60 years, probably. So lots of stuff has happened in a little bit of time, and I think it's just going to keep going that way. So that's all about biotechnology. If you have any questions, let me know.